Hello, so this is a video that Hello, so this is a video. So yeah, if Yo, what's up? Yo.
Hey,
Hey, how are you doing?
I'm okay. Always could be better, but
Health is
huh?
health is getting better.
No.
Yeah.
No, things gone better with it.
Well, at least it seems like you're okay enough to have a session at least.
Yeah. I mean, you know, what else can I do? I have a special X-ray tomorrow for my small intestine.
An X-ray for it, or like an
Yeah, it's
MRI?
a special X-ray. It's supposed to be more invasive and
Uh-huh.
stuff, and it's quicker than doing an MRI and everything, setting it up, because my gastro wants to see what's up.
Mm-hmm.
He says that if there's at least a blockage or something, it'll be there. So. Okay. Well, let's see. So what question were we on?
We were on, I think, like 21 or 22.
Okay. Yeah. So let's keep going over it, I guess.
Mm-hmm.
Yeah.
Yeah. I was looking through some of my notes here to look at the different types of polymerase. Because the question is asking which enzyme transcribes 5S ribosomal RNA.
Should be RNA polymerase 3.
Oh, yeah. I was looking for what they did in my notes, but I guess I could just look them up.
I mean, that's my guess. Yeah.
Well, that's a good guess, man.
It was correct?
Yeah.
Oh, wow. Okay. Well, I need to know why it's correct. So
Exactly. That's what
at least it was a good guess, but I don't know why.
Yeah, that's what I'm looking at now. Okay. So I'm going to oh yeah, I'm going to share my screen because I'm going to use the whiteboard.
Yeah. Bitch.
Okay, apparently, so I'll write what the different ones do here. So RNA polymerase 1 apparently synthesizes ribosomal RNA.
RNA polymerase 3 synthesizes what again?
Ribosomal RNA, like not
okay.
specific.
I see.
And let's see, product, location. Location is in the nucleolus. Polymerase 2 synthesizes mRNA and SN RNA, which I think is small nuclear RNA.
Yeah, it's supposed to be small nuclear. Huh.
And then RNA polymerase 3 makes the 5S ribosomal RNA, as well as the transfer RNA.
Okay.
Okay. Yeah, so RNA polymerase 1 transcribes ribosomal RNA, but not the 5S ribosomal RNA. And yeah, so you can kind of like write down what these do from what I wrote.
Oh, okay. Well, I have it over here on my thing. Over here.
What?
This is like the little mini thing that we made for it and stuff.
What thing?
For RNA polymerase 1 through 3.
Mm-hmm.
Yeah, so 1 translates rRNA, 2 mRNA, 3 tRNA.
Oh. You can like kind of like put that by your webcam so I can see what you're saying. You're saying like we went over this before?
Yes, we've done it before. I was trying to find it in the notes.
Okay. So yeah, 3 in addition to tRNA, also 5S.
Be 5S RNA?
Yeah. 5S rRNA, like ribosomal RNA.
Yeah.
And then in addition to mRNA, the RNA polymerase 2 also synthesizes snRNA. You like my plants? I got a bunch of plants.
Oh, awesome. What type?
I know what some of them are. Some of them I don't know. And yeah.
Okay. So RNA 1 and 2 are in the nucleolus, and then the last is the nucleus, right?
Oh, so no. Sorry. That's so only 1 is in the nucleolus. The other ones are in the nucleus.
The other ones are in the nucleus?
Mm-hmm.
All right. Well, let's continue then.
Mm-hmm.
All right.
Let's see.
So
RNA
23.
Yep. RNA
Huh?
polymerase II promoters are located on the blank side of each transcription unit.
Transcription unit. It'll be 5.
Oh wait. Okay. Yeah, I know we didn't do number 22, but it seems like we did something close to this question because I remember you answering that.
Oh.
All right. Oh, yeah, whatever. But yeah, the 5 side, aka upstream.
Yeah, because 5 goes upstream.
Mm-hmm.
And if you're
why is
promoting something, you want it to keep going, so you don't want it downstream.
Yeah, but if it was like a repressor, it would still be upstream. All the regulatory sites are going to be upstream.
Oh, okay.
Because, you know...
So promoters or repressors, no matter what?
Yeah, because essentially it's
Okay.
the thing that is... It's a switch that turns, like, expression on or off. It's going to have to be before the thing that you turn on and off, right? The switch will be before.
I see.
What's the N and C terminal referring to?
The N and C terminals?
Yeah.
Sorry. You're gonna have to refresh my mind on that one.
Okay, so DNA, RNA, there's going to be 5 end and a 3 end.
Mm-hmm.
But for amino acids or a peptide, polypeptide, instead of there being a 5 and 3 end, it's going to be an N end and a C end. Because for an amino acid, there's the amine group, which is the N terminal, and the carboxylic acid, hence amino acid, that'll be the C terminal.
Okay.
It's going to look like this, basically. So I just drew an amino acid right here. So this is the amine group, hence amino acid, which is the N terminal. This is just R. It's just, you know, for the side chain, which depends on, you know, whichever amino acid it is. And then the carboxylic acid group is the C terminal.
Okay.
Okay. Let's see. The RNA sequence is... Wait, what? Okay. Because we did this one, too.
And then this one was going to be, hold on. It was AAUAA?
Yeah. I remember us doing this problem, which is weird. Alright. Because I remember saying, like, I don't really know how you would memorize that or know that. Besides just knowing it. So, okay, you know it, so that's good. Alright. Yeah, this is a good question. What's the function of the poly A tail on most mRNAs?
It should be to hasten the degradation of the mRNA.
Well...
Or prevent the
Yeah.
degradation by the exonucleases?
Yeah, so why would it be exonuclease? Why not endonuclease?
Because it's breaking it down, which means it has to break it down from the outside, not within it.
Well, it could, well, why? Why from the outside?
Well, mRNA is attached, no? So, I mean, poly A tails are attached, so it would need to be broken down from the outside.
Yeah, so it's attached to the ends, right?
Yeah.
Like the 5 cap is on the 5 end, the poly A tail is on the 3 end, so it'll prevent exonucleases, which only work on the ends, but it won't prevent endonucleases, which work inside. So yeah.
Okay. Minimum number of tRNAs that a mammalian cell can have? 61?
So these numbers refer to specific. All right, so the total number of codons will be 64.
And for each codon, a tRNA exists?
So, and 3 of these are for stop codons. So 61 are for actual, amino acids.
Okay.
But how many amino acids are there?
$23?
Somewhat close. 20.
20, okay.
Yeah.
And
And
that's the amount of tRNAs exist?
yeah.
That's the minimum number?
Yeah, because the tRNA is a thing that recognizes the codon with its anticodon and attaches the amino acid and makes that amino acid, polypeptide chain.
Got it.
So that's something called degeneracy, which means that there's more than one codon for an amino acid. Because there's 61 codons for amino acids when there are only 20 there are only 20 amino acids. So of course that means more than one codon will code for the same amino acid.
Got it. Okay. Okay.
Let me ask you this: why are there 64 codons in the first place?
I don't know.
So that's just like in terms of total number of possibilities. So how many nucleotides is in one codon?
Four? Three?
Mm-hmm. What's that?
3?
Okay, good. And
Yeah.
how many nucleotides are there, could there possibly be, total? Let's
5.
say Yeah. We're not going to count the whatever the uracil one.
Oh, so 4 then.
Yep. And so there's 4 possible nucleotide combinations and 3 slots, right? For, let's say, for any given codon, right?
Mm-hmm.
So this can have 4. Sorry. So this can have 4 possible nucleotide. This can have 4 possible nucleotides. This can have 4 possible nucleotides. So this is just 4 to the 3rd power, which is 4 times 4 times 4. 4 times 4 is 16. 16 times 4 is?
Oh my god, 64.
Yep. So that's how you get that number.
The greatest variability, difference one codon specifying the same amino acid occurs. Would it be in the 1st and 3rd nucleotides of the triplet?
So this is the wobble thing that we talked about.
Uh-huh. Oh, so it'd be the 3rd one then?
Yes.
Okay.
Yeah. And that's because the 3rd position for the tRNA is like loose, like it's not as strict.
Got it. Okay.
Yep.
Okay. Cool. What is probably responsible for driving the conformational shift that is required for the release of ELF to initiation factor from the initiation complex? Would it be between C and E?
So this is going to be one of those things that a previous, okay. Take a look at number 11.
Oh wait, just give me a second.
Yeah, sure.
Alright, I'm back.
Mm-hmm. Yeah, take a look at number 11.
Okay. Okay.
So, do you remember what?
Well, it'll be the what do you call it? Amino acylation of tRNA.
So, so for 11, there's more than one correct answer. And
oh, the other was going to be A, right? The release of the polypeptide.
Yes. So that must mean that B, D, and E all require GTP.
Yeah, so that means that E is going to be the correct answer, right?
For
28. Or it'll be just B.
yeah, it'll be B.
All right. All right, what is the result of a nonsense mutation?
Oh, I should also write
E.
what E LF is. It's oh wait, sorry, it's not ELF, it's EIF. It's eukaryotic initiation.
Oh, factor.
Mm-hmm.
Okay.
So that'll help you remember it. So these things basically work to mediate translation initiation.
Yeah. Oh, okay. For 29, I'm thinking D.
Oh yeah, yep. We talked about that last time. Yep, it's a premature stop codon, right?
Uh-huh.
Completely jumbled. That's funny, because that kind of sounds like nonsense, right?
Mm-hmm.
A change in a single amino acid. What type of mutation is that?
Missense?
Yep. Okay.
That was a change in the single amino acid, right?
Yeah.
Okay. Okay. Hold on. Okay. Usually, mRNAs containing nonsense mutations only translated once before being recognized and selectively destroyed. The process whereby the faulty mRNAs are destroyed is answers D and D are correct. Splicing.
Well.
Or is it
Splicing
nonsense-mediated decay?
Yeah.
Yeah, that was my first guess, of course.
Mm-hmm. What was splicing again? What was splicing again?
It's like when an mRNA is like, what do you call it? Doesn't it like detach and then like put itself somewhere else within a?
Mm-hmm. Yeah.
Yeah.
It's when they when you take the introns out and you splice the exons together.
Yeah, introns out splice the exons together. Yes.
Yeah.
Okay. Then that makes sense. All right. A eukaryotic translation complex during the elongation stage is represented by the diagram below, which are the following alleles.
And by the way, splicing, like the word itself, means you're joining things together.
Okay.
So, yeah, like I kind of think about it when they edit videos, They take like seams and they splice them, connect them together. But yeah, so if you remember that it's the joining, you will know that, you know, it's like the only thing that we join together by taking the introns out are the exons. But yeah.
Got it. Okay, so this is represented by the diagram below. Which of the following labels is not correct?
So yeah, let's go one at a time. So is A the location of the mRNA cap structure?
Yeah, that's fine.
The 5 cap, yep. B, this direction here is the direction of ribosome movement.
Well, this means that it's moving upstream, right? No, downstream.
Mm-hmm.
Sorry. But everything's supposed to be moving upstream, no?
So.
Or during elongation, it should be moving downstream, right?
Well, nothing's gonna actually move upstream. That's why you kind of call it upstream, because it's like, you know, if we consider the DNA or the RNA in this case to be a stream that flows, right? It's gonna just flow essentially downstream. So we talk about upstream as things being ahead of the gene. So the only thing that's going to be upstream is going to be like the regulatory sites.
Yeah.
But yeah. So is B, you know, which is going whatever downstream, the direction of the movement of the ribosome?
Yeah.
Yep.
C is the anticodon of the tRNA molecule. That should be correct, right?
Mm-hmm.
Yeah, I think D is where it's wrong.
Let's see. D is the 5 end of the tRNA. Oh, that's a good question. Because tRNA is going to be upside down here, right?
Yeah.
So let's go to like, so okay, I drew that over here. Where the hell? Whoops. Wait, oh, yes. That's saying, okay, so for D, right? That's going to be true, because for a tRNA, this part, or rather, like, okay, so this part is the one, is the side that, or the end where the mRNA is, I'm sorry, mRNA, what I'm talking about, where the amino acid is attached.
Yeah.
And basically, whenever you attach something, whether it's nucleotides or for tRNA and amino acid, it's going to attach to the only site that it can attach to, which is going to be the 3' end, because that's where that hydroxyl group is.
Yeah.
So this over here is the 3' end, and this D part is the 5' end for tRNA.
Got it. So I was correct, right?
Oh, no. So that So D is correct, as in
Oh,
that. So that's the 5 end.
so then if all of them are correct, then E is obviously going to be the wrong one.
Yeah. So E is saying that that's the carboxyl terminus of the growing polypeptide chain.
Mm-hmm.
But it's going to be the N terminus.
Ah, yeah. Okay. Okay. I got it. All right. At which level of protein structure or interactions between R groups, amino acid side chains most important? Should be D.
Wait, I just wanted to draw. Damn it.
Maybe D.
So okay, wait. This is happening in whatever the ribosome. The ribosome. So this is like the polypeptide chain, and this is the next tRNA. And what it does is, so maybe I should just put a circle there, because okay, this is like, the next amino acid, and this is the next amino acid that's going to add onto this chain. So essentially, what it does is it attacks that carbonyl group, the nitrogen. So
Okay.
yeah. So that's why you need, the C terminal to be. That's why the C terminal leaves, because the N attaches. Okay, sorry. So let's see. At which level of protein structure are interactions between R groups, amino acid side chains most important?
I was saying probably E.
Both B and C. Let's see. Well, okay. Why don't you tell me what examples of what primary, secondary, and tertiary structures are?
Primary is obviously 1, secondary is 2, tertiary 3.
Well, yeah, but what do they mean, right?
Mm-hmm. Well, primary structure is like the amino acid just like itself, but with no changes, and then the secondary is like slight changing.
Is what changing?
Right. Like it probably slightly changes.
Okay, so these are like. So the primary structure, right, is just going to be a linear chain of amino acids in a polypeptide.
Mm-hmm.
So that's going to look like this, like NH2, let's say like R1, this is like the carboxylic acid group. And since they attach like this, right, the N attaches to that carboxyl group over there. So this over here, here, let's see if I can find a better picture. Peptide chain. So here we go. So primary structure. structure. Okay, so, all right, this could be good. Okay, so primary structure is just the sequence of amino acids. So that would look like, it would basically just look like this. So this is basically, you know, amino acid 1, amino acid 2. And here is just a single amino acid over here. Mm-hmm. So essentially they you know, two of them just they bind to each other. So this isn't a single amino acid, and when two of them bind through a, you know, dehydration synthesis, then it'll look like this. So the primary structure is just that. It's the amino acid sequence. Now, secondary structure is when, as we zoom out, that sequence can interact with itself by hydrogen bonding with itself.
Okay.
Okay, here, this is like from like a OneNote notebook that I had from one of my MCAT students where I kind of drew it out even better. So here we go. So primary structure, right, is the amino acid backbone, aka dipeptide bonds and disulfide bonds. That determines how a protein will fold. Because as we go on to the secondary and tertiary structure, we're talking about proteins folding. So the secondary structure is a spatial arrangement of amino acids near one another. So there's two types. There's an alpha helix, which forms, you know, a helix, like a kind of coil. And this is a side view. So essentially what we have is this amine group, which is, you know, nitrogen with a hydrogen or two hydrogens attached to it, or three hydrogens. Hydrogen bonds with the oxygen of carbonyl group. That's just, you know, down a couple of amino acids down. And so it makes this helix shape. So that's the side view. But if you look down on this axis, like straight down from the top, it looks like this. Meaning all the R groups are outside of the helix. In other words, the R groups, their interactions don't matter for an alpha helix secondary structure. The other type of secondary structure is called a beta pleated sheet.
Yeah.
Yeah. And so essentially, you know, you have this is an amino acid, amino acid, amino acid, amino acid. There's a beta turn and it goes, you know, in the opposite direction. That's why it's anti-parallel. And you're still going to have these hydrogen bonds between the amine hydrogens and the carbonyl oxygens. And yet again, the R groups are not playing a role in the secondary structure. It's just hydrogen bonds between the amine and carbonyl groups. All right. Now, tertiary structure is a three-dimensional arrangement, okay, and it's just one subunit, and it's mostly held together very loosely by hydrophobic and hydrophilic interactions. So this is kind of an example of one. I kind of just drew it like that. And so here's like kind of like an ionic interaction. Here's a disulfide bond. And their drawing is probably going to be better. So here, you see that secondary structure, you have the beta pleated sheet, alpha helix. And for tertiary structure, it's a three-dimensional folding pattern due to side chain interactions, aka the R groups. And quaternary structure just means that there's multiple subunits. So tertiary structure is just one subunit, but if there's multiple subunits, then it's going to be a quaternary structure. So that's various subunits associated together via hydrophobic interactions, ionic interactions, hydrogen bonding, polar interactions, etc. So quaternary structure isn't always there. An example is myoglobin, which is like hemoglobin, but hemoglobin has the subunits. Therefore, hemoglobin has a quaternary structure. Myoglobin doesn't. It's just one subunit. Yeah. So like here's like another kind of way that you might see it drawn, where, you know, these are like alpha helices, and the arrows are the beta pleated sheets, just showing you the direction that it's going in, and, you know, they're anti-parallel. So three-dimensional arrangement, okay? Alpha helix, beta pleated sheet. All right, so what do you think about 32 then?
Should be the tertiary structure.
Yeah. Yeah. So, you know, if you are taking notes or something, you should write secondary structure is just hydrogen bonding within the molecule, and tertiary structure is like interactions between the side chain groups, the side groups.
Primary is what again?
Oh, primary is just the amino acid sequence.
And then tertiary?
Tertiary or secondary?
No, tertiary.
Oh, tertiary is the three-dimensional folding of the protein based on interactions between the R groups.
Okay.
Here's some more drawings about them. You see that? So you see where I wrote primary? I just wrote like an example of an amino acid like chain. So I have like alanine, glutamine, valine, threonine, asparagine, proline, glycine. It's just amino acid like chain. And then secondary, I drew an alpha helix and beta pleated sheet. And then tertiary, it's combination of those, right? And then I kind of drew like, you know, I drew them better here using my colored pencil and stuff. Alpha helix and the beta sheet right there, the arrows. And then beta turn. Yeah. Okay. Yeah, that's very important to know. The primary, secondary, tertiary, and stuff. Let's see. 33. Which interaction is most important in enhancing the solubility of macromolecules in water?
Wouldn't it be H bonds?
Yeah,
Okay.
that's the only one that would be hydrophilic. Yeah.
A triglyceride is formed when 3 fatty acids join a glycerol by a.
Oh yeah, remember we had a question on that.
Yeah. Should be a catabolic reaction, right?
I mean, where do you get that from?
Sorry, whoops. Condensation?
Yep. Which is also known as
Dehydration synthesis.
All right, and then the reverse of that is
hydrolysis.
Yep. Now, catabolic is a more general term to mean breaking down.
Yeah, using a catalyst and stuff. Yeah, I got it. All right.
Well, things will always use enzymes as catalysts, but it's called kata because I think it's Greek for down, and the opposite is anabolic, which is up. So when you're
building up.
Yeah. So hydrolysis is an example of a catabolic reaction, and condensation is an example of anabolic. Yep.
What is the primary structure of protein referred to? D.
Yeah. I was going to say in your notes for primary structure to write backbone. That's like the best, word to use.
No, I have that written down here.
Mm-hmm.
Yeah, I knew that one.
Yep.
The presence of a polya tail in an RNA molecule indicates that...
Oh, wait. Sorry. It's the amino acid sequence of protein. Damn it.
Wait, it is?
Yeah. Because that's exactly what I wrote. I'm sorry. The peptide bonds that form the backbone are involved in it, but it's always the same. The peptide backbone is always going to be this over here. But the identity of the amino acids depend on the R groups. So the primary structure is going to be, that's why in my other notes I wrote it as like alanine, valine, the actual identity of them.
Got it. Okay, that makes sense. So the presence of a polya tail on an RNA molecule indicates that B.
All right, so this one I didn't talk about the order. I know I talked about the 3 things that happen to get from RNA to mRNA, and when I wrote those 3 things, I wrote them not thinking about order, because I always said splicing and then 5-cap and then poly A tail. So that could be confusing because they're asking about the order here. So the order of it is actually going to be, so I should have wrote it like this: 5-capping, then poly A tail, and then the last thing is splicing. So yeah, I wrote it like not thinking about the order of it, so I usually write splicing on top, so that would confuse you for this question, which requires you to know the order.
Yeah.
So now that you know the order, what do you think?
wait, no, never mind.
Yeah.
Sorry. Probably E then.
Yeah.
Yeah, no, that's correct.
Mm-hmm.
Okay. Next one, following statements about nucleosomes is false.
I'll be right back. I'm just using the bathroom. Okay. Which of the following statements about nucleosomes is false?
B.
Wait, you said B?
Yeah,
Well, do you remember what nucleosomes were?
but when E.
So okay, what are nucleosomes?
I'm not sure.
Okay, they're a unit of 8 histones with DNA wrapped around them. It looks like this. It's also known as an octamer because it's 8 of them. So we have 8 of these spheres, which are the histones, and DNA. The DNA is wrapped around, 8 of them, and that is a nucleosome.
So then probably that they're only found on mitotic chromosomes.
Yep. Good.
Amino acyl tRNA synthetase. That's an enzyme. Let's see. D.
Synthesizes tRNAs that recognize stop codons. Well, that would be RNA Polymerase 3, right?
Oh wait, yeah.
So we didn't go over this. So what it does is it assists in coupling an amino acid to its tRNA.
Yeah, that was my next one. It was gonna be A. B. Sorry.
Yeah. Wait. Yes. Good. Yeah, I kind of have it. Whoa. In that, I could take a picture of that and send that to you.
Okay.
But yeah.
Which Which parts of amino acids are involved in peptide bonds? In a peptide bond? The side chains.
So this is an example of a peptide bond right here, and this is like making a peptide bond.
Mm-hmm.
So it's something from this. And
oh wait, it's C. It's C. Carboxyl group of the one amino acid and the side chain of another.
Close. But since a peptide bond can exist regardless of what the side chain is, and this is an example of a peptide bond, it's like the backbone, essentially. And so it's going to be between this group of...
Oh, the amino and the carboxyl?
Yeah.
Okay, so A.
Mm-hmm.
The main function of tRNA, so transfer RNA, and cell physiology is to A.
Sorry, I was just drawing this for you. Yeah. All right. C, we have an amino acid here, an amino acid here, and then I kind of put this box in green for the OH group here and the H group here, which make H2O, so that leaves to form a connection between that amine group and this carboxyl group. So that's what you get over there. And now they're connected. These two things are now connected. And a water leaves. So dehydration, synthesis. Alright. So, okay. So that's for 39. Amino group of one amino acid, carboxyl group of the other, right? Alright. 40. So
it's A.
Yes, transport amino acids to be used in protein synthesis. What would B be? Or if you can just replace the word
rRNA. Sorry.
You can just replace the word carry with has. Actually,
it's rRNA.
Forget it. I was just gonna say like what has codons.
tRNA? DNA?
mRNA?
Oh, okay.
Yeah. And then tRNA has the anticodon.
Yeah.
Okay.
And then the next question is the anticodon as part of tRNA, which I knew.
Yeah.
That's fine. Since the two strands of the DNA molecule are complementary for any given gene, D isn't correct because it can only be transcribed 3 to 5, right?
Here. This is going to be relevant, what I have here.
Is it gonna be A? Not A. Sorry. I mean E. Misspoke.
Oh, well, if only one strand carries the genetic code, why would the RNA bind to the strand that doesn't have the code, right?
Yeah. Okay.
So A and B are mutually exclusive, meaning that they both cannot be true.
Mm-hmm.
And that's like, you know, could be always like a strategy to use if you don't know the answer, because if you see any other ones like that. But generally speaking, like, all right, so which one would be true then, A or B?
B.
Yes. So there's, you know, so this is an example. Coding strand is the one that has the code. The template strand is the one that's complementary to the code. And it's called a template strand for a reason, because that's what's used to make the RNA. Because if you use the template strand and then you polymerize an RNA strand based on that, it's going to be complementary to the template strand, which means it'll be equivalent to the coding strand. So basically, like here, you know, whatever, it's double stranded. We separate the strands, and the template one we synthesize the RNA from, and it's gonna be identical to the coding strand. But I wrote here only. This is the only DNA strand that's used to make the mRNA.
Okay.
Yeah, so this will be this. Make sense?
Mm-hmm.
Alright. Which of the following is not part of or an activity carried out by the ribosome?
Peptide bond formation.
Well, where would a peptide bond form then?
Oh wait, no.
So here is you know, the peptide bond kind of forming over here.
Codon recognition.
Well,
or splicing.
Where would the codon?
Splicing.
Yes, because that happens before the RNA is mRNA, right?
Yeah.
Good. And the mRNA has the codon, and the ribosome recognizes the codon. And the ribosome has an A site, a P site, and a what site?
E site.
Good. And that's where, you know, it brings in, you know, the tRNAs and makes the peptide bonds. But yes, splicing happens way before any of that stuff. It happens still in the nucleus. Alright. Okay, now phospholipids are formed from...
Would it be B?
Carbohydrates, phosphate, and 2 fatty acid. Well, think about a lipid, right?
Yeah.
It's kind of a distinct group from proteins and carbohydrates, right?
Yeah.
So it shouldn't contain any carbohydrates, unless it's like a
Oh wait,
Glycer-
I remember this. Never mind. It's gonna be D.
Yes, good. So remember for lipids that there's always going to be that glycerol backbone.
Yeah.
So like
I remember now.
yeah. So if it was 3 fatty acids, it would be a triglyceride, right?
Mhm.
But for a phospholipid, it'll be 2 fatty acids and one is a phosphate with a polar head group. That's why, you know, it's used in like the lipid bilayer, because one side is towards the hydrophilic part of the cell and then the other is the hydrophobic part. That's like the special thing about phospholipids.
Got it.
All right, what about 45?
I'm between C or D, but I'm thinking C.
Oh, okay. So, wait, you said between C and D?
Mm-hmm. But I said that I think it's C.
Okay, yeah. It's going to be C, and something that may help you remember that is, because I see this type of question on the MCAT a lot, which is basically like let's say a certain type of DNA. Okay, so we basically established that between G and C, there's 3. Oh, let me do this. There's 3 hydrogen bonds, and between A and T, there's 2, right? So let's say we raise the temperature for some type of DNA. And let's say we have DNA like 1 and DNA 2, right? And we raise the temperature to like, I don't know, 100 degrees Celsius or whatever. DNA strand 1 survives, DNA strand 2 doesn't. And a common thing, I see this type of question on the MCAT a lot. A common question would be like, why does DNA 1 survive and DNA 2 doesn't? And the answer would be DNA 1 has more G and C, has more guanine and cytosine in its DNA's whatever strand.
Got it.
Yeah, I also have like a drawing of that here. Here, do you see that?
Yeah.
So G and C, 3 bonds, A and T 2 bonds, and then here's a graph where, wait, there's the graph. Y-axis, it's kind of hard to see because of my handwriting, but it's y-axis is percent of GC base pairs.
Mm-hmm.
X-axis is temperature, and that line means that as you raise the temperature, the percentage of G and C base pairs, like basically it can only exist at, like, the higher the percentage of the G and C means that it can survive at higher temperatures.
Got it.
Yeah.
Alright, so snRNAs. Nuclear RNAs. No, they're not translated. Not any of that. D?
Yeah. The small nuclear RNAs can bind to specific sequences at intron-exon boundaries through complementary base pairing.
Okay. Next.
What would C be? What would this be?
Oh, introns.
Good. Good. Okay.
Please
Oh,
refer to
alright.
the visual
This is a
photo to
good
show us
diagram.
of pollen fashion. Oh, damn. Okay. Oh wait, okay, I think I can do this. Okay. The 5' end is gonna be A.
Good.
So that's the answer to 50. 49, the answer is gonna be E.
Good.
The figure choose a letter pointing to a box circle surrounding only a nucleotide base. It's gonna be C.
Okay. So
And then
it's weird because like D.
D or C could be the correct
Yeah,
answer, right?
because D is like pointing to the box which has
Yeah.
base
A
in it.
nucleotide bond in there, but
Yeah.
I guess they're trying to trick you there. I guess only because they said only a nucleotide base. So, in 47, it's gonna be B.
Well, 48 is going to be C or D. Because they say
Yeah, C.
choose a letter pointing to box or encircled gray highlighted area. Oh wait. I guess you make more sense here, but the answers say C or D.
Yeah. And then 47, the answer is gonna be B.
Yep. Alright. So I would say before your test to go over specifically, I'll tell you which questions. We'll go over all of it, obviously, but definitely know this number 11. Like, know which ones require GTP hydrolysis and which ones don't.
Yeah.
Because this is, like, pretty specific.
Yeah.
Let's see.
So can we just do a speed run really quick of the questions?
Oh, yeah, sure.
And I can just, like, answer them and you just tell me if I'm correct?
Mm-hmm.
Over here. Which of the following? So for number 1, the answer is going to be B.
Uh-huh. And what would that monomer be that it would break down into?
Glucose.
Yeah.
2 is going to be A.
Uh-huh. It can also be another one.
Or the Tata binding protein scheme.
Yeah. Only in eukaryotes, yep. What did the EIF stand for?
Eukaryotic initiation factor 2.
Yeah.
Purines are characterized by 2 rings, while the pyrimidines are characterized by 3. Or is it 1 ring and 2 rings?
2 ring and 1 ring.
2 ring and 1 ring. Okay.
Yeah.
Got it. I remember. The phosphate group of a nucleotide is usually attached to the 5 carbon of a sugar.
Yep.
What
What would be at the 3 end?
The poly A tail.
But
Right?
like eventually, but generally, like on a nucleotide, right? Which is just one of those guys. Because a poly A tail would be on the mRNA.
Yeah.
Because like what you're saying, like they're saying phosphate group on the 5 end, but you could also say the 5 cap.
Yeah.
But on a nucleotide, it's the phosphate group. The 5 cap is like all the way at the end for mRNA, right?
Yeah.
So while the poly-A tail would be on the 3 end of the mRNA, like the entirety of it, on the actual nucleotide itself, which could be DNA, which can be RNA, it's just on a single one. Just like phosphate is on the 5 end, the 3 end would be what?
Phosphate?
Well, the phosphate will be on the 5 end, right?
Oh, wait, sorry. No.
The OH group?
Oh, damn it! I was my next guess.
Yeah.
I wasn't like the third row of my guesses. Okay. So
Okay.
5 is E.
A and B. Yeah, okay.
DNA molecule. It's negative charge. It's gonna be the what do you call it? The phosphate groups.
Yes. And like, you know, that's what the phosphate group looks like right there.
Yeah. So 7 contribute to the thing. Oh, okay. H bonds?
Oh yeah, so this was like the pi stacking thing.
Yeah.
So while it's true that the bases are the base pairs are hydrogen bonded to each other.
Hydrogen bonds? Yeah,
But
it
it's
wouldn't
like
be like that, so it should be van der Waals, right?
Yeah, which is a type of whatever. It's gonna be van der Waals and hydrophobic interactions.
Yeah, it's E. I was just about to say that. Okay. The information encoded in DNA resides in...
The keyword there is stacked, if you see that, by the way.
Oh, okay.
Think of pi stacking, maybe even write down like pi stacking.
Got it.
To help remember.
Okay.
Mm-hmm.
Information encoded in DNA resides in the DNA base sequence.
Yeah, this is like the question about primary structure, but that's for a protein. The backbone doesn't have information, so that's why the peptide backbone isn't the primary structure, but the side groups sequence is. And this is just for the DNA equivalent of that. All right?
9 is E, release factors.
Yep.
10 is... That's 16 chromatin. Heterochromatin?
And what's the...
I mean, euchromatin, sorry.
No, you're right the first time.
I was right?
Yeah.
Okay.
And euchromatin is a loose version.
Yeah, okay. Leuchromatin is loose, heterochromatin is a stronger version, yes. Compacted. Alright, got it. Of the following steps associated with translation, what does not involve the hydrolysis of GTP?
Yeah, this is the one to memorize.
Yeah.
Because this one, yeah, because we didn't go over these particular steps as in depth.
Yeah. So this one is going to be, well, involve the hydrolysis of GTP. A is definitely true. Right?
Well, A is one of the answers, so it doesn't involve hydrolysis of GTP.
Uh-huh. And the other one is the amino acylation of tRNA?
Yeah, so both of those do not involve GTP, which means that the other ones do involve GTP. So this is the question to like memorize. Like, don't just memorize that A and C are the ones that don't require GTP, memorize that B, D, and E require GTP.
Yeah. Okay. 12 is going to be the Eukaryotic.
You said that last time. I don't know what I don't think the Eukaryotic is anything. They're kind of taking a karyotype, the first part of that, and the mirror part.
Yeah. Oh wait, is it telomere?
Yeah.
Sorry. Okay.
Yep.
Got it.
Because it's at the tips.
Yeah.
And it's
This
heterochromatin, which means it's not expressed.
Yeah.
If it was in the middle, what would the answer be?
Centromere, which is the next one.
Good. So you see how they're like taking words? They take telo and then they put phase to mix it up with telophase and karyotype. Yeah.
Yeah.
13?
Oh, that was such centromere.
Yeah. Good.
14 is going to be glucose and galactose.
Good. What was this? Glucose and fructose? What was that?
That glycogen?
Well, glycogen would only make glucose, right?
Yeah.
So this is sucrose.
Sucrose, there we go.
Like sugar, like basic whatever sugar, right?
Yeah.
But yeah, glucose and galactose is lactose. Good.
Yeah.
If you forget, which I doubt, you get the word lactose is in galactose.
Yeah. RNAs. So they should be A?
Actually, everything besides A.
Yeah, it's E. Damn it. Yeah. I knew it. Yeah. Alright. Blank help RNA polymerase recognize promoters should be the, what do you call it? Transcription factors.
Good.
Just transcription factors.
Good.
17 is upstream from the site.
Yes.
All three major types are processed before they mature. They are derived from the precursor. And so precursor RNAs are called pre-RNA.
Yeah.
rDNA clusters gather together to form the should be the nucleolus.
Mm-hmm. What type of polymerase is in the nucleolus?
It should be only RNA polymerase 1.
Good.
20 modifications of nucleotides of pre-RNAs made should be post-transcriptionally.
Yep.
Ribosomes are made of both proteins and lipids. No, proteins.
And?
Yeah, lipid and lipids, right? I mean, no, not lipids. Sorry, rRNA.
Yeah.
So it's E.
Mm-hmm.
Yes. What enzyme transcribes that? RNA polymerase 3.
Yep.
Okay, 23 is the 5. Side.
Mm-hmm.
24 is A.
That's just a weird one. Just memorize.
Yeah. 25 is B.
Mm-hmm.
26 is A.
Mm-hmm. What's the 64?
Hold on. Hello? I almost. It's in a few minutes. Alright, bye-bye. Okay. So this one is going to be the third nucleotide of the triplet.
Yep. That's called
wobble codon.
Yeah.
Okay. Was it probably responsible for the GTP hydrolysis?
Yes.
Okay. Result of a nonsense mutation is the premature termination of the polypeptide chain.
Mm-hmm.
30 is going to be the non-mediated decay.
Mm-hmm.
31 is E.
Mm-hmm.
32 is going to be C because of the folding and all that stuff.
Well, what did the secondary depend on?
H bonds.
Yeah.
And then the first one is just amino acid.
Yep.
Which interaction is most important? Enhancing solubility. That'll be hydrophobic and H bonds. I mean, H bonds. Sorry.
Yeah.
Hydrophobic.
Mm-hmm.
I was thinking of that last question.
Mm-hmm.
Triglycerides form 3 fatty acids, throwing up glycerol by condensation reaction.
Yep.
Primary structure refers to the amino acid sequence of the protein. Presence of poly A tail indicates that what do you call it? None of the answers of that is correct.
Good.
Which of the following statements about nucleosomes is false? That they contain, they're found in the euchromatin.
What was nucleosome?
It's what do you call it? DNA that's wrapped around by euchromatin.
Wrapped around histones.
Wrapped around histones, yeah.
Yeah, and the thing is, euchromatin and heterochromatin, they still have histones and nucleosomes. It's just that euchromatin has the nucleosomes spread apart.
Oh, I remember this one. So they're not. It's false that they're only found on the mitotic chromosome.
Yeah, and that's a good question because when you think of nucleosomes, you think of them being condensed, which happens in mitotic, like chromosomes, but they are still in non-mitotic chromosomes. It's just that they're spread apart more.
Yeah. 38 aminoacyl tRNA synthetase. It's going to be B.
Yep.
Parts of amino acids are involved in the peptide bond. Should be the carboxyl group of, the amino group of one amino acid and the carboxyl group of the other.
Yep.
Main function of tRNA in cell biology is to transport amino acids to be used in protein synthesis.
Yep.
Anticodon is part of tRNA.
Yep.
And so 2 strands are complementary. That's B. Only 1 strand carries the genetic code.
Yep.
Which of the following is not an part of or carried out by, so it's splicing.
Yep.
Phospholipids are formed from a glycerol, a phosphate, polar head group, and 2 fatty acids, so
Yep.
D. Adenine forms with 2 H bonds, and guanines form with 3.
Yep.
So yeah, C. snRNAs is D.
Yep.
And then this one is 47 is B, 48 is C, 49 is E, and then 50 is A.
Yep.
Okay. Alright, I'll keep reviewing it.
Yeah. How do you feel about these?
The genome questions. There shouldn't be much on the genomes, but yeah, I'll keep studying these two.
Oh yeah, yeah. Like, you know, these questions. Like, do those also, okay?
Yeah.
The transposons and stuff. Okay.
Alright. Will do.
And remember, like the GTP questions.
Yeah.
All right, just like have it all like completely memorized, so you can't go wrong. Oh, and know which ones are purines and pyrimidines as well.
Yeah.
Okay.
Okay.
Is that it?
Good. Yeah.
All right. Hope you feel better, and good luck, man.
Thanks.
All right. Take care.
Alright.